All right. Um, so almost everybody has submitted the field guide. A couple people have not. I do accept late assignments, but after a certain time point, I have to start taking points off for being late. But I'm usually allow a grace period, so um, in general, that's how I run all my assignments. So it's best that you communicate with me if you know it's going to be late, but it's better to turn it in late than not turn it in because you'll still get points for it. All right, so in lab, you're going to continue on this week on Friday with more of your project prep. And then next Monday, Duncan will be out of town, so I'll be joining you in lab and hang out with you and help you. So, and that's the plan for that. And then next Thursday, a week from tomorrow, will be the next exam. I don't think it's going to be as big as the last one. And I don't know that there'll be a take-home portion of it either. The first exam had a lot to do with methods and, 3 PowerPoint slides full of methods of how to sample and run experiments. So that was a pretty big idea that I wanted you to think about before you started your projects. But now you started them, and this set of information that you're getting is not as big. However, I am planning, I think, well, let me run this by you. What I thought would make more sense is to kind of look at what I have left to talk about. And so we have 2 more exams. If I have to wait one a little bit longer, probably the second one rather than the final. Usually the final exam, students prefer a little bit shorter exams for the final because you're so busy with wrapping everything up and finals and everything, all your classes. So that's kind of how I've done it in the past. What do you think about that? Okay, so that's my plan. But once I get a more solid draft, I will talk to you more about next week's exam. I'm almost done grading the take-home assignment, so I'll let you know when that's done. Okay, so I wanted to talk to you a little bit more about endophytes before we get into some of the other concepts of microbe-plant interactions. And so we were looking at this idea that, they have to come from somewhere. How do they get to be endophytes? Most scientists believe that the soil environment is the primary method for how microbes get in. So they're typical soil microbes that can colonize the root surface, hang out with the plant, and then some of them manage to get in and persist. And that can be through perhaps the root hair entry, or it could be through cracks and abrasions of the roots. The soil is a pretty abrasive environment, so the roots can get slightly damaged. And with the scale, how tiny microbes are, you can imagine it might not be so challenging sometimes for them to get in. And so that's definitely a method. And we also looked at the idea of entry in by the stomata of the leaves, which is certainly has been documented in many different plants. And so bacteria and fungi, here you're seeing some fungal hyphae getting in through the stomata. And bacteria, I've shown some images of bacteria getting in that way as well. So that's well documented. And then some, when they become endophytes, they start to form such a tight association with their host plant that they can migrate into the developing seed as that is forming and actually get transmitted to the next generation that way. And now they're realizing that could be a very important way to set up that baby plant in the seed for success when it does germinate, because it's coming with microbial partners that the mom plant had, so to speak. And so from that perspective, it does make sense. It's similar to when you were born, you were immediately well colonized by microbes from your mom and then from your family unit shortly thereafter. So certainly some strategies that can make them end up being successful endophytes, we also covered the concept that they can move once they get in. They don't have to stay put wherever they gain entry. They can move through the plant body. Sometimes it can be through the vascular tissue, the xylem and phloem, as the plant has a full internal highway system, so to speak, of ways to transport water and minerals and sugars after photosynthesis is done. And so having that vascular tissue as an internal highway system could be a very nice route for them. Here that just demonstrating with a rice plant how the migration from the root into the above ground portions and into the leaf tissue. And then we looked at some ideas here with Streptomyces in the seed, so embryo tissue that's talking about the seed, inside the seed. And Streptomyces are soil microbes. They're very, usually almost any soil on the face of the earth, you're going to find Streptomyces present to some extent. And so they're just super well adapted to soil, but we're finding also that some of them can go endophytic and make it all the way to the seed. So I think we left off here. Looking at the idea, this paper was trying to show that not only did they end up in the seed, but they seem to have grown in population through the length of this study. So they were able to show that it's definitely persisting and seems to be well accepted as an endophyte by the plant host. Here's some other images looking at root tissue. So cortical cells are just a different way to talk about root cells. And they're trying to show Streptomyces form petite hyphae. So they're trying to show some of the hyphae showing up in there as well. So certainly the more they look, the more they find endophytes in every plant. They now suspect every plant is well colonized, both externally and internally, and that they are playing a role that we were largely unaware of for many millions of years as they co-evolve with each other. Don't forget about aquatics. Aquatic microbes can also, aquatic plants can also have associated microbes with them. So this is Azolla. This is with the naked eye on the top right here. It's a petite little cute plant that tends to float on the surface of water systems. And it's technically a fern, aquatic fern. And if you look carefully, primarily at the leaf tissue in some of the valleys and cavities of the leaf tissue, the bottom left image is cyanobacterial cells. Anabaena tends to string together in cells that look like a pearl necklace and hang out there. And so the cyanobacteria, of course, is also capable of doing photosynthesis, but its big benefit for the plant is that it fixes nitrogen. And so some of that fixed nitrogen can be given to the plant. Can you tell what this is? Yeah, it's kind of hard to tell though. I had to like triple look at it because I found it and they're like, oh yeah, there's a frog. And I'm like, where? But it's amazing how well colonized. This is all Azolla, even though it looks pink and purpley and reddish. That happens when it's under intense sunlight. It tends to produce more pigments to protect itself against intense sunlight. So sometimes you'll see patches of Azolla that aren't the lush green color, but still really beautiful pigments. So definitely a win-win. Aquatic plants have been less studied than plants in the soil systems, but when they look there, they're also finding endophytes and epiphytes. So, and then here's a wacky one. This is a virus inside a fungus, which is inside a plant. So now we have this, kind of like those nesting dolls that you see. It's kind of that way. And so this was an interesting paper published in Science, which is a huge journal. If you get published in Science, that's like just amazing. And they were looking at this idea that what's going on with the virus, what happens if you get rid of the virus and just let the fungus hang out with the plant. And they were looking at that idea. So the thermal tolerance, this idea, if you put the plant under heat extreme, what would happen? And so if they get rid of the virus, so it's called curing, you can cure microbes of viruses, in lab experiments, you can do that. And here, that's what they did, and then they put the fungus in the plant, and they could see that the plant did not survive high heat. But if they kept the virus in the fungus and had them go endophytic, then yes, the plant survived. So there's some kind of interaction between the virus and the fungus, and then, what's going on with the plant to allow it to survive the high heat. And so this is just a mock-up trying to show that 65 degrees, that's really warm temperatures. That's amazing. So this idea that when they looked at some of these environments and saw the extreme heats in desert environments that some of these plants were surviving, they started to look more carefully, and that's what they found, a virus in a fungus in a plant. And then here's another wacky one. Here's a bacteria in a fungus in a plant. And so it's a fungal endophyte. And when they peered inside the hyphae, they found bacteria hanging out inside the hyphae. And this is Dr. Arnold's lab out west, and she's been doing a lot of work with endophytes as well. And looking at, in this case, they were looking at the production of indole 3-acetic acid, which is a plant hormone. Phytohormone means plant hormone. And they found that when they took away the bacteria and the fungal endophyte, that we didn't see the same growth promotion as if you had them with it. So when they got rid of the bacteria in particular, they could show that it wasn't producing the indole acetic acid itself, but somehow when you combine the bacteria and the fungus, so they're starting to wonder if the interactions between the bacteria inside the fungus was somehow stimulating the production of IAA, perhaps by the fungus, which then gives that IAA to the plant, which stimulates plant growth. So again, the more they look, the more they're finding these complex interactions of microbes associated with plants. So it's an interesting field of research right now. This was published quite a while ago, actually, 2008, but I like the title, More Than 400 Million Years of Evolution, and some plants still can't make it on their own. And so again, stressing, this is Rodriguez and Redmond. They're a married science team out west and have done a lot of work with plant microbe interactions. And they are, in this paper, they were looking at endophytes inside plants. But basically, yeah, over 400, plants evolved on Earth around 450 million years ago. And they are suspecting that right from that time period forward, you had associations with microbes, and the endophytes are part of that as well. And so just like they're finding out with us, with the human microbiome, that we need our partners, our microbial partners, to function correctly, they're finding that to definitely be the case with plants as well. So these endophytes, the more they look at them, they're saying, what are they doing for the plant? And they're realizing that sometimes the endophytes are producing the same chemicals that the plant can produce. And so, of course, plants are famous for producing all kinds of secondary metabolites, chemicals that are typically in response to some kind of environmental challenge or situation. And that's why we get so many of our drugs for human use through studying plants and their chemicals. Well, then they started looking at endophytes and realizing, oh, guess what? All these endophytes are also capable of producing these really cool chemicals, same ones that the plants are doing. So this is probably the most famous example that we have to date, Taxus, which is the genus name for yew trees. And this is the story of an endophytic fungus that hangs out in the bark of yew trees. And the drug, the chemical paclitaxel, which they've nicknamed Taxol, which might sound vaguely familiar to some of you if you know anyone who has cancer, specifically breast cancer. And so this is a drug that is incredibly successful. It can put breast cancer patients into full remission for years if they keep taking it. And so it's highly successful. And so of course, when they isolated it from yew trees initially. And when it became a billion-dollar drug, they're like, of course they kept chopping down yew trees and grabbing the bark and extracting the Taxol. And some of the plant biologists are going, uh-oh, if we continue at this rate, we're not going to have any yew trees left. And so some microbiologists said, well, could it be maybe an endophyte in the yew tree that is producing Taxol? And if so, that would be a better situation for the plant because we could just work with the endophyte in a lab environment, grow it up in a big vat and get it to produce Taxol and then extract the Taxol and sell it and keep the yew trees alive. And so what they did was they went to the bark tissue of a variety of different trees and came back to the lab. They started hunting for endophytes and they found one. And it was a fungal endophyte. The name of it was Pestiloteopsis microspora. And so, yes, the yew tree apparently can make Taxol, but so can the endophyte fungus. And so this was hugely good news for plant biologists, knowing that if they could just work with the fungus and leave the yew trees alone, they could still make this very successful drug and help a lot of people survive breast cancer. And so definitely a great win to keep everything, the yew trees from going extinct, essentially. When they started working with this fungus, they realized that it's really easy to manipulate genetically. And so this most easily transformable fungi means that it's very good at taking up DNA and working with it, like turning on the genes that it gets. So this horizontal gene transfer idea, they're saying this fungus is just really good at it. And so that makes some of the scientists think that maybe the plant was doing this Taxol production first. This fungus became endophytic, and then it acquired some of the genes, the genes for Taxol production from the plant and started producing it as well in partnership. And so that's the current hypothesis on how the fungus ended up making this drug. It's a pretty complicated drug if you look at it. They don't know how to make that synthetically yet. I don't know if they ever will. And so they still get it from natural sources from the fungus. So a huge success story. And so this is an example of the plant making this paclitaxel, the fungus now making it. But sometimes they're finding that when they study some of these endophytes, they produce chemicals that the plant doesn't seem to be able to make. So these are unique chemicals that the host plant is unable to do. And so you can imagine this might be really good for the plant if the endophyte is producing a chemical that is beneficial to the plant, then the plant doesn't have to do it. It's just relying on the fungal endophyte to serve that role. And so this could be super good for the plant host. And then they're also finding in lab studies that some of these endophytes they've identified, they can produce a lot of different chemicals. And so they're very good workhorses for the plant in the sense that many of these chemicals may be certainly benefiting the plant host. So again, if you think about it, if a microbe goes endophytic, first of all, that is dicey because the plant has to be able to accept that and not kill it, right? So if you are getting microbes into your body, your immune system, and plants have an immune system, they have to be able to say, okay, yeah, I'll accept you in as a friend. So if they're going to co-evolve with each other, the idea that if the endophyte is persisting, it's probably doing something beneficial for the plant. Otherwise, the plant might have just gotten rid of it. And so that's why they're so excited about looking at endophytes, because it's such a tight association between the endophyte and the host that they suspect the endophyte has some services for the plant that we can take advantage of in the sense of novel, unique chemicals that we can use for human health or animal health. So lots of looking going into like anti-cancer drugs, and all kinds of, they're looking for new antibiotics that our current microbes aren't resistant to yet. So lots of ideas out there for chemical discovery. And here, this is just taken from a paper trying to look at, what are these endophytes doing for the plant? And certainly we'll talk about nitrogen fixation in more detail, but that's technically those are endophytes that are doing that. A big idea is phosphate solubilization. I'll talk a little bit more about that too. So phosphates in the soil, plants have to have it in high doses. And it can be very hard for a plant to get access to it because a lot of phosphate in the soil is insoluble. And if it's insoluble, most plants can't get easy access to it. They need phosphate dissolved into the water of the soil. And microbes help with that. This idea of plant growth promotion in general is looking at the health of the plant. What can it do for the plant? And so siderophores are, so microbes can produce siderophores. These are chemicals that latch onto iron and chelate the iron. And so it's a way to make sure that the plant is getting enough iron. And then many of the endophytes are known to produce plant hormones too. So auxin is a plant hormone. And then the idea of using endophytes as biocontrol agents is another interesting idea, looking at whether or not they can protect the plant. And so these are some of the ideas that they're thinking about in terms of how could endophytes be helping the plant defend itself or just reduce disease, plant pathogens. So looking at production of the siderophores again, the volatile organic compounds, which can be inhibitory pathogens, hormones. Induced systemic resistance means that some microbes are able to stimulate the plant's immune system in a full body way, systemic, through the whole body. And so you can see that the entire plant starts to become resistant to the pathogen starting to attack it. Antimicrobials, of course, different chemicals that can directly attack the pathogen, but then other chemicals that can help the plant like ramp up its immune system, get access to key nutrients so it can, have a strong physiology and hopefully be able to defend itself. So lots of ideas are being studied. Here they're looking at different lead compounds with a focus on quorum sensing disruptors. And so again, biocontrol agents, but they're zooming in on quorum sensing. Can they mess with the quorum sensing of pathogens by using endophytes? Can we get the endophytes to do that? And so this quorum sensing idea, they're trying to show that they're disrupting it. In could be a couple of different ways. Maybe the endophytes are breaking down this chemical signal that they're using for quorum sensing, or maybe they're stopping the synthesis of it. Well, lots of ideas are being investigated on how they might be able to use endophytes to mess with the communication system of pathogens trying to attack the plant. And then we'll talk about this later in the semester, but I just wanted to put this here as a little note of the idea that they're looking at endophytes to help with remediation of toxins in the environment along with the plants. And so that idea is called phytoremediation, where you're using plants, but really it's not just the plants, it's the plants and their suite of microbes that are capable of breaking down the toxins. So pretty much, at least most of the studies I've read, it's not just the plant doing the remediation. It's this partnership between the plant and its associated microbes. And so one study that they looked at was the idea of I guess this is toluene degradation. And so what they were playing with is the idea of using endophytes, mixing in endophytes to the plant and seeing if the bacteria could go endophytic, go inside. And then they were looking at the idea of if we mixed like a cocktail of bacteria, including one that can break down toluene with other bacteria. So the idea is, we know plants have endophytes. And we know, some bacteria can break down toluene. So could we mix the bacteria that breaks down toluene with the native endophytes in this plant? And get them to interact with each other? Could there be a way to transfer the gene for toluene breakdown? So conjugation, we talked about, that's part of the horizontal gene transfer. And so could we get the endophyte that we know can break down toluene to donate some of the plasmid or toluene breakdown into the native endophytes, and then inoculate that with the plant and then see what happens. And when they ran that experiment, you can see here that there's different doses of toluene from the front image. The front plants are the control, where there's no toluene, and then all the way up to 500 milligrams per liter. And then you can see that no introduction of the microbes, your control. They didn't do so well when you started to even introduce 100 milligrams of toluene. But if you look off to the ones that were getting, mixed together, so these last two rows there, you can see that, especially VM1330, that mixture worked really well. And so they're looking at this idea, which is exciting because now they're realizing, hey, we might not have to like force a partnership, because what if this endophyte that they know can break down toluene, what if that it doesn't really hang out with that plant and they don't really ever form a partnership? But we know that this plant has native endophytes. Can we just kind of get that trait over to the native endophytes and have them take over? And so they're looking at this idea, which is great, because then you don't have to have a perfect match with, finding an endophyte that works well with those plants. You can just allow horizontal gene transfer to take place. So nice idea. It's I kind of young stages of research on this, but I think there's a lot of potential, because we know horizontal gene transfer is like a thing. Microbes do this. And so it kind of makes sense to attack it from that perspective, to try to find better partnerships to break down all the toxins that we have put out in our environment. So very nice idea. This last idea here, this is Carrie, who is currently a professor in the indoor egg program here at Northern, but she was my grad student. And she did her thesis research on the Ram's Head lady slipper near Munising, Pictured Rocks National Lakeshore. And this orchid is either extinct or endangered in most places of the world, but for some reason at Pictured Rocks, it is flourishing. They estimate, Adrian Bozic, one of Dr. Robertus's grad students, did a very extensive survey of the Ram's Head lady slipper and were shocked to find over 2 million, they estimate over 2 million individuals of this orchid. So there's something about that habitat that's allowing this orchid to flourish. And so Carrie was a good friend of Adrian and helped Adrian with her research. And then Carrie and I started doing research on this with the idea of, well, why? Why is it so well adapted to that environment? And we were suspecting the microbes underground, perhaps, or endophytically, were playing a role in that. And so she started investigating that and discovered that, yes, there's certainly endophytes that were present. She extracted the DNA and got the sequences back for ITS, internal transcribed spacers. That's how you zoom in on fungal DNA. And she was able to sequence and identify several different endophytes, including a couple that she's named here. And then she was studying them more carefully about, well, what could they be doing for the plant? And they were finding, she was finding that these endophytes were definitely helping the plant with nutrient acquisition and nutrient breakdown. And basically it was helping the plant, feed itself essentially. And so this is just a excerpt from the conclusion of her thesis, but she's making the point that we need to better understand, especially with rare plants like this that are endangered, looking at their endophytes and seeing what we can do to preserve these orchids and keep them alive in these different habitats. So one of the issues is, of course, climate change. Is this orchid still going to be able to handle the pictured rocks area with climate change, and could those endophytes help the orchid adapt? So really nice project looking at endophytes of a very rare orchid in most areas of the world. So I like this quote. You know that there's all kinds of destruction of forests, especially tropical forests. And that's sad in and of itself, but now they're realizing you have all those epiphytes and endophytes that are lost when you get rid of all those trees. And they're really excited about especially tropical forests because of the sheer diversity of plants in that area and the strong competition between those plants. They suspect endophytes are playing a critical role in helping plants in those types of environments. And so, which is kind of leading to the idea of a lot of these endophytes could be super helpful for chemical discovery, using them for drug discovery. And so just from that perspective alone, it's a sad thing to think about with all those trees and all those endophytes getting wiped off. Okay, on that happy thought. So sorry, but unfortunately, it's a reality. Okay, so epiphytes and endophytes. Lots of exciting research going on in especially endophyte research that's really taken off. And the more they look, the more exciting it gets in terms of they're realizing all the different things going on that those endophytes are providing for the plant host. Okay, but let's continue on. The next slide set I think is available in EDUCAT with mycorrhizal fungi. So those of you that are taking Plant Phys or have had Plant Phys, you're going to get this again. So this will be an easier section, I guess, of this course, this part of the course for you because you've had this. But definitely we got to talk about it. We got to talk about it in this class because this is a huge concept in terms of plant microbe, many huge concepts here in terms of plant microbe interactions. And this is, I mean, all three of these topics, mycorrhizal fungi, nitrogen fixing, and plant growth promoting microbes, they are all just pivotal for the survival of plants. And actually, they point to mycorrhizal fungi as the kind of the bridge organism that helped plants make the switch from aquatic oceans to terrestrial life. They suspect through many careful genetic studies, fossil records, they're pretty confident in saying that mycorrhizal fungi were there as the plant was shifting from the ocean onto terrestrial environments, and that they help the plant deal with that huge and dramatic switch. So we're going to be zooming into the soil for this, and particularly the area right around the roots, which is called the rhizosphere. So when you look up the definition of the rhizosphere, it's kind of nebulous, but basically they say something like, it's that area around the roots that the root can influence. And then they say something like, it's a few millimeters, in diameter or something like that. But basically, we're looking at this idea that root systems, vast majority of plants have their roots in soil. Roots are continually sloughing cells off as they move through the soil. You have damage, some cells get sloughed off. You have chemicals getting exuded by the roots on a minute by minute basis. So it's a very enriched area of the soil. If you compare soil with roots compared to just bulk soil that has no roots and look at the chemical profile, very, very different. And so the presence of the plant and what it's doing with its roots, both physically and chemically, really impacts the soil environment. Here are some ideas of what is going on in the rhizosphere, looking at, both physical and chemical. And then the idea that microbes really love to be around roots because it's a wealth of chemicals that they can use for nutrition. And so if you take soil samples right by roots versus bulk soil with no roots, you'll see huge differences in microbial populations. Microbes love to be around the plant roots, very high populations compared to root-free soil. So we'll talk about some of these ideas that are summarized here in the table. So there's this phrase that's been coined for quite a while in the scientific arena. They call it the rhizosphere effect. And that's basically just what we were talking about, the plant's influence on the soil system. And again, going back to the idea of the plant roots moving through the soil as they grow, they're sloughing cells off, they're excreting all these nutrients. So they often use the word leaky, and they use the term exudates. There's things being given off, all kinds of things, but certainly also organic compounds, which means a lot of microbes that break down organic compounds are typically right there by the roots, waiting for those compounds and using them for their nutrition and ways to get their ATP. So definitely plant roots affect microbial composition and populations and diversity in the soil. Absolutely. And in general, there's a couple of general big picture ideas you can say. Gram negatives tend to be more abundant compared to gram positives. Not always. This is kind of a rule of thumb, but it's not 100% black and white here. And so you tend to see rapid growers though, because of all the pretty lush nutrients that are being provided. And definitely, like I said, higher populations of microbes compared to root-free soil, definitely. So initially, I mean, they knew about this for decades and decades. They knew that roots can do all of this. But they initially thought that that was pretty much it in terms of what the plant is doing to impact microbial communities in the soil. And now, more recent, within the last couple decades, more recent research is showing that plants are much more capable of influencing, for example, they can communicate and encourage beneficial microbes to their roots. And so they can recruit certain microbes over others. They can certainly defend themselves against plant pathogens. They're not at the mercy and just totally unable to defend themselves. They have sophisticated mechanisms to try to defend themselves and immune systems. That took a while to figure out too, and they're still trying to figure all that out. And so plants are way more dynamic than they initially thought. They thought they were just kind of sitting there and they were at the mercy of everything that came along. And now they know that they can be very particular about what microbes they recruit, what microbes they inhibit, and that they will deliberately enter into partnerships through communication between, vastly different kingdoms of life. You have plants versus microbes, very different, unrelated organisms. And so they've been certainly studying the other way around, how are microbes affecting plants? And of course, they've definitely been zooming in on the plant pathogens, rightfully so, but we're also starting to catch up on what's happening with the good guys, the beneficial microbes. And so I'm going to focus more on that for this section. And so auxins and gibberellins are plant hormones that a lot of co-evolved microbes can also produce. So they are definitely plant hormones, but the microbes make them, the microbes excrete them to the plant takes them up and uses them, and it's a beneficial thing in most cases. Some plant pathogens can also do this though, but a lot of beneficials will do this and in a good way. Plant pathogens do it in a more selfish way for their own gain. But anyways, beneficials also can produce different organic compounds and vitamins that can be beneficial to the plant. They can, the more they look at this idea, the plant defense mechanism, the more sophisticated we're starting to see the interplay between beneficial microbes and plants in terms of helping the plant defend itself and working with its immune system. So very interesting mechanisms at play here for the ability of beneficial microbes to help the plant fight off other microbial pathogens or even insects or worms or whatever is attacking them. Definitely increase mineral nutrient availability. I'll talk a little bit more about that. So you need to have lots of microbes in the soil in order for the plant to get access to the minerals they need. And I'll talk more about that. Microbes, we know, are very good at recycling nutrients. They're capable of breaking down all kinds of organic matter, getting it into its simpler forms, recycling it in the different environments, and that definitely helps the plant because then they can get access to some of those breakdown products. And the microbes are just really good at breaking down compounds that are insoluble, that the plant can't use, and making them soluble. So bacteria and fungi have great enzymes that they can excrete out there and attack insoluble precipitated chemicals. So this is a mock-up trying to show that idea that you need to have chemicals dissolved in the water in order for the plant to easily get access to it. So it's very, very challenging for a plant to get access to something that is precipitated out of the water in the soil or attached to the soil particles. So here they're trying to show the different balls are perhaps different minerals. So when you look at the soil system, that's, when minerals are attached to soil particles. And the minerals have to come off the soil into the water of the soil so the roots can take them up. Roots really have a tough time doing that. It's virtually impossible. There's a few exceptions with some of the minerals, but by and large, they have to wait for those minerals to become soluble. And so microbes have to be doing that. And so without lots of microbes in the soil, you have plants suffering. You can have all the minerals in the world in the soil system, but if they can't get access to it because it's attached to the soil or precipitated, then the plants are stuck. So if you're taking soils class, or we talk about this in Plant Phys too, when you look at the soil system and the plant needs all those minerals, but the minerals are attached to the soil, you got to get them off the soil somehow. Those minerals have to come off. And so there's a process called cation exchange that helps with that. And I'm not going to really go into detail in this class on that. But basically it's a chemical process that allows minerals to get kind of bumped off as water flows across the soil. You have minerals in the water that can literally bump off other minerals that are attached and do an exchange process. And so cation is referring to positive charges. So we're talking about things like protons or potassium or magnesium. So anything that is positively charged, there's this idea that they can substitute. They can kind of bump off another cation off the soil and take its place. And that's like a continual process that can occur with proper water flow through the soil. So that's great. That's definitely going to help the ones that get bumped off. They'll go into solution and the waters of the soil, plant takes them up. The other idea though that I want to focus on is the microbes. And so the microbes, through their enzyme action and their abilities to essentially break down minerals and rocks. You know, fungi are great at that, so are bacteria. They have all kinds of funky enzymes that allow them to solubilize. So there's a lot of words that you see in the literature. They'll say, oh, they solubilize the minerals, they weather the rocks. They'll use that term. They'll say mineral dissolution. It all means the same thing. Rock degradation. So basically all of these terms, you see that a lot in the literature. Just trying to say that the microbes are really good at getting those minerals off the soil. And then that gets dissolved in the water and then the plants take them up. So again, you have to have a good populations of microbes in the soil in order for that to occur. And we've already learned that plants are definitely well colonized and that this is a partnership. In many cases, we have a lot of beneficial microbes. And so they are definitely calling it the plant microbiome, just like they call, you know, our microbiome, like, you know, the human microbiome, there's a plant microbiome. So this plate was a soil dilution that I did of an experiment I was running, and you can see trying to point out the diversity here where every single different looking colony is a different species. There's actually quite a bit of Streptomyces here too. All these kind of grayish, whitish, blackish, or pinkish are Streptomyces bacteria. And then the endophyte idea, that's a leaf tissue with endophytes coming out onto the tissue culture medium. So when we think about the plant growth promoting microbes, a lot of what they've been zooming in, you know, they've done a lot of research on the root area, the rhizosphere. And that's actually been longer research going on compared to the endophyte research. I like this quote, root systems which absorb nutrients and are directly exposed to soil microbes are functionally analogous to the animal gut. So they're saying they have a lot of interactions going on, similar to how our gut is well colonized by microbes. You know, our intestinal tract is well colonized by microbes. It's kind of like this is their gut out in the soil environment, and you have similar interactions with their rhizosphere microbes and their microbes that have co-evolved with the plant. So that brings up the idea of mycorrhizal fungi, which are technically, some people call them endophytes. I disagree. Endophytes, I hold to a more strict definition of endophytes live internally, totally internally to the plant. We'll find that mycorrhizal fungi can go internal. Parts of them are internal, but the vast majority of the fungus is external. So kind of a special case. They belong in a group of their own. So we'll continue that on tomorrow and look at them more carefully.